Hello, everyone. Today, I am truly honored to introduce a research paper published in iMeta titled, "The Combined Influence of Microbiome and Soil Environment Contributes to the Chemotype Differentiation in Atractylodes lancea." Previous studies have categorized Atractylodes lancea into three genotypes, Ma, Na, and Sa, as well as to chemotypes, MSA and HBA. The highlight of this research lies in its clarification of the environmental factors influencing the genotype and chemotype of Atractylodes lancea. Genotypes exhibit genetic differentiation into Ma and Sa along the gradients of altitude and climate. The chemotype differentiation is not genetically determined, but is instead shaped by soil environmental conditions and the microbiome. In this research, we collected a total of 187 wild and cultivated Atractylodes lancea from 16 populations across 5 provinces nationwide. Among them, 109 plants were subjected to genome resequencing. The remaining 78 rhizomes were analyzed for volatile oil content, its sequences, and endophytic microorganisms. Resequencing results indicated that Atractylodes lancea from 5 different origins could be classified into two genotypes, Ma and Sa. The Sa genotype is exclusively found in high-altitude regions, whereas the Ma genotype is exclusively distributed in low-altitude regions. The genotypic differentiation is influenced by altitude and climate. However, genotype alone does not fully determine chemotype. We conducted a diversity analysis of the rhizosphere and endophytic bacteria and fungi in Atractylodes lancea from various habitats, and the findings indicated that the rhizosphere bacterial and fungal diversity were significantly greater than those in rhizome endophytes, and the microbial community composition in the rhizosphere soil closely resembled that in the bulk soil. Among the rhizosphere core bacteria, Burkholderia and Streptomyces are exclusively found in the MSA chemotype, whereas Pentabacillus and Sphingomonas are solely present in the HBA chemotype. In the rhizomes, the endophytic core bacterial communities of Atractylodes lancea from both chemotypes exhibit similarity. Functional prediction of the core bacteria in the rhizosphere was conducted using PyCrust-2, revealing functional differences in the microbiomes between the two chemotypes, HBA and MSA. We cultivated seedlings of two genotypes in the native soil NMS from MSA producing and the native soil NHS from HBA producing, respectively. The findings revealed that the NMS enhanced the accumulation of atractylone and atractylin, whereas the NHS boosted the accumulation of heinzol and β-eudesmol, independent of the genotype. Meanwhile, we conducted the aforementioned cross-planting experiment again, with the modification that the indigenous soil underwent sterilization. The results revealed that the sterilized indigenous soil exhibited a comparable promoting effect on the volatile oil content of Ma and Sa to that of the non-sterilized soil. However, the impact of sterilized soil on the composition of volatile oil is less pronounced than that of non-sterilized soil. Subsequently, we discovered that the core Streptomyces, exclusive to MSA, facilitates the growth of Atractylodes lancea, whereas the core Pentabacillus, which is only found in HBA, inhibits its growth. Additionally, endophytic core bacteria were found to promote the growth of Atractylodes lancea. Further analysis indicated that two strains of Streptomyces unique to MSA could inhibit the accumulation of heinzol and beta-eudesmol, while promoting the accumulation of atractylone and atractylin, thus favoring the formation of the MSA chemotype. Conversely, Pentabacillus specific to HBA enhanced the accumulation of beta-eudesmol, contributing to the formation of the HBA chemotype. Although the endophytic core bacterial compositions of the MSA and HBA were similar, the functions of different strains varied, which might account for the further chemical differentiation. High levels of potassium and phosphorus suppress the accumulation of atractylone and atractylodin, while promoting the accumulation of heinzol and beta-eudesmol. Elevated temperatures and high rainfall primarily strengthen the interaction between rhizosphere bacteria, fungi and atractylone, whereas altitude intensifies the relationship between heinzol or beta-eudesmol and the microbial community. The structural equation modeling results reveal that altitude and climate can exert both direct impacts on the chemotype and indirect influences through genotype and microorganisms. However, genotype does not have a direct effect on the chemotype. Similarly, soil nutrient composition not only affects the rhizosphere bacteria and fungi but also directly influences its chemotype. The study reveals that altitude and climate are the driving factors behind the genotypic differentiation of Atractylodes lancea. Yet genotype alone does not determine chemotype. Instead, microbiome and soil environment are the key drivers of chemotype differentiation, with microorganisms exerting a particularly significant influence.